delightsome morning, and to all the Canisius, a tranquil evening. As we all know, I'm sorry again. Swayamdipta is our alumni. He has fetched B.Tech degree from NIT Durgapur in the year of 2010, and he has earned PhD from the University of Alberta and is a former NIH postdoctoral fellow. Presently, he is enacting out the role of an instructor at the University of Texas. So, without wasting further time, I will request you, Swamdipta, please begin with this wonderful conclave. He with that of three F's: flows, fills, and forces. So we are really enthusiastic to hear a mysterious tale of synergy at nanoscales. Thank you so much.
Well, thank you, Lisa, for the kind introduction. Can everyone hear me all right and see my cover slide?
Yes,
Awesome.
clear.
Awesome, fantastic. So, just a quick correction. My first name's pronounced as Swayamdipt. The last A is silent, so I'm not Swayamdipta. I'm Swayamdipt. Such a pleasure and honor to be back here. It's all good. Everyone makes that mistake. So I'm very delighted to be back. This the second time I've been invited to speak at NIT Durgapur since I graduated in 2010. The first time was in 2018, right after my PhD, but that was an invitation from the Alumni Association, not the SAI Cell per se, and that was in person when I was visiting India. So this is very different, but it's always an honor and pleasure to be able to do that. So thank you to the organizing committee for inviting me today. I want this to be very informal, so do not hesitate to interrupt, to stop me at any point of time if you want to chime in, if you have any questions, or if you want more clarification. If there's anything else you want to share, please unmute yourself and interrupt me. I'm certainly not one of those types who want this to be very formal or certainly don't mind being stopped at any point of time. That's what the idea is. I believe this has to be interactive to be fully beneficial to the student. So please do not hesitate. I certainly encourage you to do that. My topic today is, as Lisa said, it's flows, fields, and forces. I tend to think there's a mysterious synergy at nanoscales, which I'll hope to show you a little bit about today. And when I say F flows, fields, and forces, it's just not playing with words and doing an alliteration on F. I'll make a case that these three indeed kind of synergize and interact and intertwine in ways that's very mysterious, with lots of open questions for scientists like us and you. And this is really a fascinating topic to explore. And by the end of this talk, I hope to convert you all to consider pursuing research in the related fields. So that's a hidden agenda as well. I'm here to recruit my colleagues and future cohorts who can kind of push forward the scientific agenda in the field, which really is exciting and certainly needs more bright young people like you all. So a very quick trajectory about me so far. These are kind of the highlights of my career since I graduated with a BTech in mechanical engineering in 2010. Those are the best years of my life that, in hindsight, I still miss. I had job offers from DRDO and ISRO, which I declined because I was never really cut out for industry. I knew I wanted to do a PhD, but I wasn't sure where or what field. Mechanical engineering is too broad. I knew I was fascinated by fluids, but then once again, fluid mechanics can have so many different sub branches as well, and I didn't really have a clear idea. Um, I dabbled with research in various shapes and forms for the next 3 years until 2013. And I was really lucky and fortunate to be able to work with some very good scientists, some here in India at IIT Kharagpur, some at the London Center for Nanotechnology, which had just recently opened doors a little while ago, in the UK, mostly at Imperial and University College London through now defunct program, which was then called the Commonwealth Split Site Sandwich Program. But I was not formally enrolled in any of in any academic program until 2013 when I started formally applying to grad schools. I applied to 12 different universities. I applied only in the English-speaking Commonwealth nations, Canada, the UK, and Australia. I did not apply to the States. I received 8 offers from these universities that's listed, and I decided to go to the University of Alberta in Canada, Edmonton, Western Canada. That was a difficult choice, but in hindsight, I made the right decision. That's what I like to think. Primarily for 2 reasons. First of all, I actually got to work at the National Institute for Nanotechnology, which is the flagship nanotech lab managed by the National Research Council of Canada, which was then the largest federally funded nanotech national lab in the world until 2020. Recently, in the last 2 years, there have been some newer facilities which are bigger in scale in some matrix, but it still remains one of the big 3 nanotech labs in the world. They had amazing state-of-the-art facilities, and I was really lucky to be able to work there. And secondly, my advisor was Professor Carlo Montemagno, who has been widely regarded as the father of bionanotechnology. And for those of you who do not know, he was a Feynman laureate. And in case you're wondering what that is, just like you have the Nobel Prize in, let's say, physics and chemistry and medicine, and if you win the Nobel Prize, you are a Nobel laureate. There's something called the Feynman Prize, named after Richard Philip Feynman, one of the preeminent physicists of the 21st century. And he is considered to be the father of nanotechnology. And this is the highest honor in the field of nanotechnology. So this is oftentimes called the Nanotechnology Nobel. And Dr. Montemagno was a laureate as well, and I was once again extremely lucky to be able to have him as my PhD advisor. Um, then I moved to the States. I was a National Institutes of Health and IH Fellow at the UT Southwestern Medical Center in Dallas. I was affiliated to the Green Center for Systems Biology, which then was headed by another Nobel laureate, Johann Deisenhofer. So I was really lucky to be able to work in a place like that. And then since 2020, during the peak of the pandemic, I've been at the University of Texas at Austin, which is the flagship campus of the University of Texas system. I am a PI of records, which means I'm a principal investigator. I'm not tenure track, but I'm considered a research faculty, pathways to independence. I am able to supervise grad students and advise PhDs, but I can only do so not independently, but as a co-advisor to someone who is a tenured faculty. Uh, I'm kind of transitioning to more independent tenure track positions. Hopefully, around this time of the year in 2023, I'll be leading my own independent lab in some shape and form somewhere, and I'll be heavily recruiting my first grad students and maybe even postdocs depending on the funding situation. So if you're a senior or if you're a junior and you plan to pursue research in kind of broadly the fields that I'm going to talk about today, I'm certainly looking forward to chat with you. If that's something that resonates, I certainly encourage you to reach out and we can talk if we might be a good fit in a year or two from now. So that's kind of the hidden agenda, and there's the reason. I gladly accepted the invitation from the SAI Cell because I wanted to kind of promote what I do and kind of pre-recruit any interested students if that becomes possible in the next year or two. All right. So this is today's plan. And this is my plan. It doesn't have to be written in stone. And I'm very happy to kind of play along with what you all want, because this is all about the audience and less about me. I visited the YouTube channel that Saisal has, and I kind of browsed through some of the wonderful talks by other eminent speakers over the last months, and some of them have stuck around for like 2 hours. While I don't have that kind of time, I am happy to stay longer than I plan. It's a Saturday morning for me, and I don't have any urgent agenda coming up. But the real technical talk, I'm hoping it'll be about 35 to 40 minutes because, as I told Lisa before everyone started filing in, I have not included any mathematical details at all. I realize that there are students from all different specialties. There are not only mechanical engineers, but people from all other fields. And there are freshmen and sophomore, and there are seniors and juniors. So I don't really know how to kind of set a level that speaks to the universality of the audience. So I decided to focus more on the physics, include lots of videos and images, and less on the math. I'm happy to talk about any mathematical aspects if you're curious. So do stop me if that's what you want to hear. But this talk, being very physical, shouldn't take longer than 40-45 minutes. I'll talk about the three apps, which is kind of my niche. This is my specialty, the intersection of the three apps at nanoscales, the kind of problems that I work on, and I'll make a case why is it important. This is not a something that lets me satisfy my intellectual curiosity, but it also has tangible translational engineering applications. So I'll certainly make a case for that. Yes, does anybody have a question? All right. Um, I'll talk about how it is done. Um, I will kind of give an overview of the techniques that the community uses to kind of give you a bird's eye view of how this field works. And that's until this why and how. It's not really any original research that I have been doing really. I'll probably give you more of an overview of what are some hot trends, what are some pressing problems that the community is working on, without necessarily touching on some of the specifics of the particular projects that I have been working on, until I come down to the what question, where I talk about what can we, meaning anybody working in this niche field, can do with it, and I hope to give you some glimpses of at least one problem statement that I've been really inquisitive and enthusiastic about over the last year or so. And then I'll leave time. I'll open the floor for Q&A. That being said, I'm very happy, as I said before, and I reiterate, to take any questions, any and all questions, and field any kind of curiosities or any inquiries you might have while the talk is in progress. So just unmute me, just interrupt me and mute yourself, and just shout it out, and I'd be more than happy uh, to address it to the best of my abilities. So while it says 15 to 20 minutes, assuming that it's an 1 hour endeavor, I'm very happy to stick around for longer if some of you want to chat or just say hello, or share your comments or suggestions about the talk, or have any kind of feedback or any ideas, or anything else to share. Everything is extremely welcome, and I certainly appreciate that. So I'm happy to stick around for longer. And I think it's less about me, more about you. You probably have more interesting stuff coming up on a Saturday evening that you're looking forward to than listening to a nanotechnology talk. But if you do, I'm very happy to, very gladly oblige. So this is kind of the plan. Does anybody have any questions, or does it sound like a good plan so far?
It's fantastic.
All right, thank you. Let's move forward. So the fascinating three apps. And as I made this case before, I'm not just alliterating and like trying to say something that sounds good and it's rhetorical, but this is something that's really at the crux of several pressing, very challenging, and very mysterious, if you will, physical problems that challenges the challenge the scientific community to kind of access some of the deepest resources known to the community to tackle them and challenge them in ways that's quantitative and that enlightens us on the physics. So I'll walk you through this. I'll probably spend 5 minutes on the slide. All it looks deceptively simple. There's a lot going on inside, and I want this to be something that you really appreciate, because the entire talk really kind of rests on this as a cornerstone. So when we talk about flows in the context of, in the context of nanoscale systems, we hardly talk about simple liquids like water or simple gases like air, although that's possible, but that's typically almost always in conjugation with more complex fluids which show highly nonlinear rheological and viscosity behavior, and highly nonlinear correlation between other macroscopic physical and thermodynamic properties, and more measurable quantities like viscosity and surface tension. And we don't exactly understand the molecular mechanisms behind them. That itself is complicated enough. It becomes more complicated when we go into the domain of what's called biological fluids engineering, and when you start um, studying fluids that's typically found inside biological bodies, biological organs, organisms, things like blood or saliva, or all kinds of hormones, all kinds of chemical molecules, biological macromolecules, which have a very diverse range of functions. And these have not only a highly nonlinear behavior, as most complex fluids do by definition, they have strong coupling with things like the topographical and topological features of the landscape that the flow is occurring in, that the landscape that it's navigating. Um, and uh, and that kind of brings us to this area called biological microfluidics or biomicrofluidics, which was my PhD topic, really, if I had to point my finger and select one level, one keyword to it, which itself is a hot field. There are dedicated journals. There's this journal called Biomicrofluidics, really, literally, which speaks to the fact that this is an entire monster in its entirety by itself, can be an independent field. When we talk about forces, as engineers of any shape and form, we know that it's essentially any kind of device engineering, any kind of instrumentation, any kind of measurement systems, any kind of analytical systems. Really, any kind of design engineering rests on accurate and precise estimation and quantification and measurement of different kinds of forces, their ratios, and how these forces can kind of sway and vary over the length scales and time scales associated with a particular problem. And these considerations become much more challenging, much more interesting, uh, much more nonlinear when we talk about mesoscopic length scales. So without going into a lot of details, when you talk about macroscopic scales, think about roads and bridges and bigger structures and buildings and skyscrapers and things like that. And when we talk about really tiny nanoscales, think about atoms and molecules, or cellular aggregates and clusters, and biological messengers, and DNAs, and even orgasmic and organismic scales. Somewhere in between these two extremes is what's called a mesoscopic domain, which itself is a very rich field. Historically, it was called mesoscopic physics. Now it's called a soft matter for reasons I'd be happy to talk about, but that would take us on a tangent that I don't really want to intentionally. I'm an experimentalist, so I work in an area called experimental soft matter physics. And if you kind of graduate onto the biological side of it, just like microfluidics has this biological flavor of biomicrofluidics, experimental soft matter physics has this biological sibling, if you will, that's called cellular mechanobiology. As the name suggests, kind of marries the aspects of mechanics with biology at cellular length scales. And the name is kind of misnomer. It has stuck around for historic reasons. It doesn't have to be exactly cellular. You can kind of graduate to bigger length scales like organ level, or even organ system, full organism level considerations. And kind of tying these two together is what's called the chemical physics of surfaces and interfaces, because as you'll realize more and more once you get into this field, and you'll probably get a little bit of glimpse today when I present this lecture, um, um, these kind of these interactions between the flows and the forces in a framework of multiple fields really occurs, really happens at several kinds of interfaces. This could be 1D or linear interfaces. This could be 2D or superficial surface level interfaces. So these could be 3D or volumetric uh, full spatial interfaces, which can also have a temporal time evolution aspect. And that's where the surfaces and interfaces become really important. And for somebody like me, I'm really interested in the chemical physics aspects. So in this Venn diagram, let me clarify this. Whatever you see in the big bubbles, like in the flows, fields, and the forces sets, these are kind of well-established fields, and there are lots of smarter people who have been kind of working on the classical problems in this field for at least 2 decades now. These are not new per se. But once we start kind of looking at the intersection, things become more interesting and things become more kind of open-ended and the territories become less charted. And this is where you have more opportunities to do research because less things are known and there are more open problems, if you will. And in the fields scale, we have, so the fields that's important for what I do. And of course, I can give you an endless list, but that doesn't really help anybody. But for this talk in context, I'm mostly interested in what's called active matter. There are certain things which are self-propelled, things like bacterial cells, for example. They have mechanisms like beating flagella or Ty4 pili or different kinds of different designs of cilia that let them transfer themselves from point A and point B, between point A and point B in space and time, which means without adding additional energy, external energy in a field where they exist, they can actually move and translate and move and change their position as time evolves. And these kind of matter, which may or may not be biological in origin, these could be purely chemical in origin as well. But self-propel matter, this is very loose and non-rigorous definition, but that would serve the purpose for now. These are called active matter, and this is something that's fascinating, and we don't understand a lot about it. Uh, Partially because they show what's called self-organization. They can kind of create patterns and they can assemble into structures which are highly functional and which can show advanced functionality that as engineers, if we are able to mimic, we can design a kind of cool stuff, kind of mimicking what nature does. And nature is the epitome, the ultimate engineer. And that gives us some cues on what we can do at the machine and device levels. And that brings us to the idea of what I call emergent phenomena, meaning order out of disorder, something that does not exist initially, but as time evolves, something that was seemingly random and chaotic can organize in ways that leads to orders and layers and layers of sophisticated orders. And once again, that's something we do not at all understand well. So as I said before, these are all kind of very broad fields, and these are huge fields, and there are established research groups, research centers, national labs dedicated to research in all of these fields. And while I work at the interface, I do not specialize in any of these per se. I specialize in areas where these things kind of start interfacing and overlapping. So when flows and fields overlap, we have these areas called multi-phase flows, things like phase change heat transfer, if you have this thermal component to it, if you add thermal fields, and the collective dynamics in living systems, things like swarm robotics, for example, rely heavily on interaction of these kind of fields and flows. When flows and forces of these kind interact, we have microscale transport phenomena. We have microreology, and we have complex colloids. And when forces and fields interact, we have things like biomedical optics or biophotonics. We have high performance imaging and characterization of advanced functional biomaterials. Some of the hardest samples to actually image and characterize. This is not something you can put under the microscope and get a picture or do an SEM or do some kind of a regular, XPS or XRD and, conclude about the functionality or composition. And that really pushes our engineering know-how and our ingenuity and our ability to think out of the box and our creativity and our resourcefulness to its very edges. And that's where real creative, interesting science happens. But when all of these fields kind of interact and overlap, this really niche small field. Let me see if I can have a good laser pointer. Let's see. Yeah, I got that working. So here, this is ME. It's a pun. It's not mechanical engineering, but it's me, meaning I and myself. So this is where I work. This is what I like to say is my niche field. And this area, this really small sliver, this tiny sliver of this scientific domain is highly nonlinear, where multi-scale domains rich in polyphase physics occur. And we know near to nothing about this. This is something I have spent the last 10 years or so studying, and like, while by no means I'm an expert, I'm probably one of the first generation of scientists studying this niche field. And this is where the fascinating 3F's gonna come in. This synergizes really nicely at the nanoscale. So none of these phenomena are really important or interesting or equally fascinating or equally open in the sense that things are more known and more well understood at large macroscopic scales. Things start getting really interesting at the mesoscopic scale that I just alluded to. And they are absolutely crazy at nanoscales where, we don't at all understand why things happen the way they do. Things are very counterintuitive. Sometimes we observe regularly, we measure phenomena that seem to defy laws of regular physics as we understand them today. There are quantum entanglements that I do not do at all. So my premise is entirely classical. I don't do any quantum mechanics, but the idea is the richness and the nonlinearity existing in this really niche field is so mind-bogglingly it's really bewildering. It's really something that lets you gasp with awe and wonder. And this is something I find really fascinating. And my talk, if there's one agenda today, is to kind of share a bit of that fascination and convert maybe a handful of you to consider research in the broad field. So yeah, that really is what I mean by the fascinating 3Fs and synergy at the nanoscale. And I'll give you more details as we keep going. But does this kind of make sense? Does anybody have any questions, or do you kind of see where I'm going with this? Do you kind of like feel a little bit of my excitement? Chime in if you like to. Okay, let me play these videos, like this. These are really nice videos. All right, so just to clarify, these are from one of my favorite labs at the University of Washington, directed by Dr. Albert Folk, who is a fascinating scientist. I have had the opportunity of speaking to him on multiple occasions. He's just a brilliant guy. And he calls this the opera and the symphony at nanoscales or microscales. And the examples that we saw are examples of what are called microfluidic devices. I'm not sure why my slides are not changing. Okay, I'll just keep going for some reason. Okay, let's see. There you go. So, what you saw were some examples of what are called lab-on-a-chip devices, like nanofluidic, microfluidic devices, which are really small. And we'll quantify the smallness of it, how small it is really shortly. But the idea is you can have this beautiful beautiful phenomena that illustrates the synergy of flow fields and forces at the nanoscale with incredible levels of control and rigor. Don't let yourself be misled to think that this is something we do just to kind of satiate our intellectual curiosity here. This is something cool, which does not really have practical applications. It does, and this is a very short, this is a very conservative list of applications of immediate translational applications of research on those kind of platforms. And we can do separations and screening and sorting of all kinds using these kind of platforms. We can create mixtures with incredible amounts of isotropicity and homogeneity that's impossible for classical systems. We can design point of care diagnostic devices that can detect things like existence of COVID antigens or not within seconds, which otherwise would take a whole day in a sophisticated pathological lab. We can have what are called the lab on a chip and organ on a chip systems that emulate the complex biophysical processes taking place inside biological systems in a small handheld system. We can simulate things like oil fields and oil rigs in a small handheld device, the so-called reservoir on a chip. We can generate droplets and bubbles with incredible uniformity, and I'll show some videos that illustrates that point. We can work on sensors and detectors with amazing R values, amazing high throughput and yield, and amazing screening resolutions. We can emulate complex chemical physics of all kinds of real world systems. We can design bioreactors. We can design digital microfluidic systems that can act as logic gates, and I'll also give you some examples. We can control chemical circuits. We can do drug delivery, biophysical analysis. We can design all kinds of biomedical micro devices that can be implanted inside the human body. For example, something that releases insulin at regular intervals for patients who have diabetes. We can have all kinds of MEMS devices, micro electromechanical systems. Coolings, things like your smartphones, your iPhones, your laptops. Right now, as we speak, have sophisticated nanofluidic transport cooling systems embedded inside their motherboard. And but for them, those would overheat and kind of stop working and completely ruin themselves within minutes. And microarrays and amplifications, things like PCR and DNA amplifications for those of you in the biological side of things would appreciate. These are all made possible and then some possible and in levels and degrees impossible at large scales via the synergy of the three apps at nanoscales. Isn't it cool? Is there somebody who thinks it's not cool? Just let me know and I'll convert you. Hopefully not. So, let's define it. So I've been talking about this mysterious nanoscale right now. How big is that? How small is that really? So this is kind of this number line for length scales, if you will. 1 meter is typically what we encounter in our day-to-day lives. This is the length scale of a adult human body, and this is the corresponding volume scale, 1,000 liters, really. And this is where most classical engineering devices and instrumentation works. Talk about roads, bridges, buildings, most of the machines, and this is where traditional engineering has been operating over the last 100 years or so. At the very extreme, you have molecules, 1 angstrom, 0.1 nanometers. We have things like viruses, COVID viruses, for example. So the length scale is about 1 nanometer to 1 microns, and the corresponding volumes would be 1 femtoliters, 1 attoliters. And this is the domain of traditional nanotechnology, nano devices. Somewhere in between, this is traditionally the domain of microfluidics, which I don't do precisely. I have more kind of graduated or more gravitated towards smaller length scales because that's where most of the fun stuff occurs these days. But this is where you'll find most of the traditional micro tests, total analysis systems, microneedles, microfilters, reactors, micropumps. Human hair is kind of about this length scale. So 1 nanoliter is the volume of 1 single strand of human hair. Uh, And this is kind of the length scale of bacteria as well. Some of the smallest bacteria could be as small as viruses, but typically most of the pathogenic BSL2, BSL3 bacteria will be kind of the same length scale here. So this probably, hopefully, puts things in context. So in the future, moving forward, when I talk about the synergy at nanoscale, we are really talking about this length scale, this volume scale, the one that's enclosed in this kind of this red rectangle. So something to kind of keep in mind. Uh, so why small, really? I have made this case already, but if you're still not convinced, this is an example of what's called a microfluidic chemostat. And this is a 1 cent American dollar coin, just that I just grabbed the other day somewhere. This is a typical nanofluidic handheld device. And as you can see, these are all kinds of fluidic connections. These can be electrical connections, all kinds of sensors and detectors working with what are called data acquisition systems. It provides us with a static chemical microenvironment that can be used as maybe a bioreactor where we can add a fresh medium and we can culture, different kinds of cells that emulates the microenvironment that you find in their niche biophysical habitat. And this device can provide single cell resolutions to study microbial population growth. And I'll talk briefly about it in the future. Uh, you know, um, this is one more example. This is an example of what's called protein crystallization. So proteins are really important for those of you who are in biotechnology or, on the biological clinical side of things. And we can generate droplets that can encapsulate and contain proteins and work and act as cargoes and act as container ships that can transport proteins from point A to point B of interest. And we can have them crystallized. We can control the timescales associated with these processes, and we can screen for optimal protein crystallization conditions. That's a marker for several diseases or like problems or like some kind of a condition that speaks to something that's not healthy. Um, this is entirely impossible in bigger length scales. And bubbles and foams, we can generate bubbles and droplets which are tunable and monodisperse. We can do that with incredible regularity and uniformity. And there are so many different applications that I'll show you a short video about. And these are kind of just a small sliver of examples of what's possible using the synergy. And I leave this to your imagination to think, what might be accomplished once you are able to do that repeatedly with high amount of faithfulness and repeatability and reproducibility, and how you can kind of interface all this to design instrumentation and devices in an engineering sense to accomplish goals otherwise impossible for bigger length scales. Let's watch a really, really cool video.
Microfluidics studies the behavior of fluids at the micro to nanometer scale. Microfluidics can miniaturize the work of a whole lab onto a microfluidic chip less than a few inches big. One thing microfluidics is great at is making consistent droplets of fluids. They can be used in cosmetics, chemistry, biology, and even as medicine. To make droplets the regular way, take a beaker filled partly with water and partly with oil, two liquids that can never mix. When you stir, droplets start to form. They are of different sizes. When using this technique at an industrial level, inconsistent droplet size can be an issue, and some of the liquid will be lost because you cannot control the quantities precisely. Now, imagine being able to control the exact size and quantity of droplets with a faster and more precise process. Microfluidic equipment can do that. For the same experiment, you need a basic microfluidic chip, a see-through slate with engraved channels only a few nanometers wide. In the microfluidic channels, you can control how you make the droplets. Put the fluid for droplets in one channel. The other fluid will pinch it from both sides and make tiny drops of fluid that collect in the pool on the right. Their size is strictly controlled. There is no waste of product, and the process is fast. You could make up to 1,000 droplets per second. Great, but why make droplets at all? Well, this miniature droplet is actually a microreactor. It can be made of any type of fluid, and we can trap what we want to observe inside. This microreactor droplet can be used for many things: cosmetic creams and lotions, supramagnetic nanoparticles, diagnostic tests and assays, mayonnaise and low-fat food with air-filled fat particles, controlled medicine delivery with biodegradable microparticles, and many more.
So isn't it cool, right?
This is a fantastic, splendid,
Awesome,
great
awesome.
splendor. So
Does anyone else have any questions? Lisa, would you mind moderating it? Like if someone raises their hand and such, I might not be able to monitor it. But if you see that,
okay.
yeah, thank you.
Okay, I
Appreciate
will
that.
inform you.
Fantastic. So does anybody have any questions until this, or does don't be shy. Come on, like there are no no stupid questions or no incorrect questions. So if you have any comments to share, just feel free to chime in. Um, so now that I have like set things in context, really, uh, you might be wondering, well, so far so good. This looks cool, and all that. This is, I see Sway ranting about this being fascinating, but how can we translate these ideas into nanofluidic devices? This is obviously not trivial. This is not something that's intuitive. You cannot really create channels which are of the order of nanoscales or even micron scale channels. It's incredible, right? I mean, you cannot go to a machine shop and kind of drill holes, or you cannot go to a carpentry shop or an electrical shop and like cast it. Like that's just not possible. That opens up an entire different field. It's called nanofabrication, and there are dedicated groups that spend their lives and careers doing this. I do not, but I enjoy the fruits of their labor and their intellectual contemplation. And I'll show you one more short video where you get an idea of how we translate this to tangible devices, where how we can build these things. So yeah, let's see.
The Lutetium Project's Microfluidics Adventures, Chapter 2. We recently published the first episode of our Microfluidics Adventures. Click here if you haven't seen it yet. Microfluidics is fluid dynamics at the microscopic scale, and fluid dynamics is the science of flows. So what we want is to make liquids flow at the microscale. How do we do that? In a microfluidic system, you often find the same components: pumps, valves, pipes, and all of these are no larger than a hair. It's basically microplumbing. So, what do we need? First of all, we need to make several different liquids enter the system. Then we need to build channels to make them circulate. Then, well, we need to make something happen. That's kind of why we built the whole circuit in the first place. So the liquids interact. We'll talk about that in Chapter 3. Finally, we need to analyze or collect the volumes we've transformed. And the goal is to make all of it hold on a glass slide, like this one. It's only a few square centimeters. And did you see all the things we want to put on it? That's what is called a lab-on-a-chip device.
Let's take an example. Let's say we want to create a microfluidic circuit which produces water droplets in oil. To do that, we're going to use a well-known technique, flow focusing. In our microfluidic chip, there's going to be a channel where we're going to make the water flow. We're also going to put two channels transporting oil, which will connect with the water channel at a right angle. The oil is going to pinch the water and fragment it into small drops, and we'll collect them in a big pool. So there, we have the outline of our circuit.
We know what we want to do. Now, how do we do it? By micromolding.
We'll create a mold based on the outline. On that mold, we'll pour plastic. We'll turn the plastic into a solid, take away the mold, and glue the plastic on a glass slide. Now we have pits where we drew channels.
Of course, when we get into details, it's a bit more complicated than that, and I can already guess your questions. First of all, how do we make the mold? We're going to use the technique of photolithography. We're going to engrave a pattern on a substrate with the use of light.
First, we create a mask. We imprint the negative of the channel's pattern on a transparent sheet of plastic. Then we'll need a silicon wafer as support, and a layer of resin which will spread on the support. If this resin is exposed to light when heated, it can't be dissolved in a solvent. So if it's lit up through the mask, the zones corresponding to the channels are the only ones which become insoluble. We can then get rid of the rest of the resin by immersing it into a solvent called the developer.
And now we have our mold. That was theory. How does it work in practice? We said we wanted microscopic channels, but if we don't make them in the proper environment, it only takes a single grain of dust in the channels to block them. This means we have to work in an extremely clean room. It's literally called a clean room. And since we're doing photolithography in there, we're not going to choose the color of the lighting at random. We're going to light the clean room in orange. Our resin is sensitive to green light. If we shined this color on it, there would be a parasitic exposure. That's why we use orange, which happens to be a color that does not contain any green. It's kind of like developing analog photos.
So here we go. We take a silicon wafer and pour resin on top. We spread it by spinning it very fast and heat it up to solidify the resin. Then we take the mask and put it in the lithography machine. We also put our wafer with the resin. We light it up, take the resin back out, heat it up, and develop it. We get rid of the non-lit up parts with a solvent. We clean it, and the mold is ready.
Now the plastic. Most of the time, we use polydimethyl siloxane, also known as PDMS. When bought, it looks like a transparent and viscous liquid, but it can easily be solidified thanks to a compound called cross-linker, which creates chemical bonds between the PDMS chains. So really, all that needs to be done is pouring it on the mold when it's liquid, then hardening it.
And that's what we're going to do. We start by surrounding the mold with aluminum. We take liquid PDMS, then some cross-linker, mix them together, and pour the mixture on the mold. The whole setup is put under a vacuum bell to eliminate air bubbles, and then we wait for about 10 minutes. Once the bubbles are gone, we take the PDMS and put it in a heated enclosure to speed up cross-linking. After an hour, we can take out the PDMS, which has become solid. We take away the aluminum and the mold and cut out of the PDMS the area around the circuit we want. Now we just need to make holes with a punch for the liquid's inlets and outlets.
At this point, we're almost done. We have our channels molded in PDMS, but they're still open on one side. We're going to close them by sticking them to glass. And the glue is nothing less than plasma.
We're going to put the PDMS chip in a glass slide in an atmosphere of extremely energetic pure dioxygen. It will make every exposed surface very reactive. All it takes is putting the PDMS on the slide for chemical bonds to form. We've just made an irreversible bonding.
So there you have it. In this video, we saw how we get from this to this. Now the question is, what are we going to...
Isn't it cool? So this is like, super complicated, and, I mean, it gets condensed in a 5-minute video, but it's probably a process that lasts for like 3 days. And as she said, you do this inside a super clean room called a clean room, literally. And you have to like dress up like something that looks like an astronaut, like a space suit, where not a single inch of your body is exposed, because that would totally ruin the cleanness of the environment. And when I started, when I graduated in 2010, there existed no academic clean rooms in the entire country in India. Since I've graduated, SENS has come up at IISc, which is probably the only class 10 clean room that exists in the country right now. I might be wrong. There might be some newer ones that have come up that I'm not aware of. None of the IITs had it. So if you wanted to do research that involved clean room and nanofabrication, you had to go abroad. And NINT, where I got my PhD, had the largest clean room, class 10 clean room in the world, probably still does. So that was very interesting. Even today, there are probably about 20, 25 class 10 clean rooms on the planet that you can academically access. And obviously, companies like Intel or AMD, the chip manufacturers, have class 1 clean rooms accessed by robots, but that's not something you can use for academic research. So fabrication is very complicated. I'll skip the slides because I have already shown you all the steps, but the take-home message here is that using that process, that very complicated process inside the clean room, we can fabricate chips like this. Isn't it cool? Like, you have a small chip that you can hold between your 2 fingers, and you have all kinds of fluid circuits going in and out. You have electrical connections, you have sensors. You can apply these fields. You can apply magnetic fields, you can apply acoustic fields, you can change chemical concentrations, you can heat it up if you place it in a environmental chamber and kind of control the pH and the humidity and the atmospheric pressure and the ambient temperature associated with it. And that's where the field part of the 3 Fs come in. And using these, you're able to ask and analyze wide range and incredible variety of questions that's otherwise impossible to ask if you do not have things like that. So don't let yourself be intimidated. I can go into more details if you like, but the idea is once you know how to fabricate and assemble things and create these embedded systems, creating these kind of lab-on-a-chip devices isn't very far away from where you start. So that was really kind of the idea of what the community is doing these days, and like the powerful tools that you have access to kind of solve wide variety of questions, and I ask a wide variety of questions that people haven't asked before, or maybe they wanted to ask but did not have the tools to address them. Now we do. So that brings me to like the last part about what we can do with all this. You might still be wondering, this looks cool, but what it is, Sway, that you are working on. And I briefly talk on one of the problem statements that I've been fascinating with for the last well, a year or two. Since my arrival here at UT Austin. And, I cannot go into specific details for two reasons. First of all, it hasn't really matured to the extent where I have published papers out of it. I have not. So, there are, I had to sign NDAs, there are mandates from funding agencies, and I cannot go into a lot of details. But nonetheless, I'll give you a little bit of idea on maybe what it is that I'm trying to do, and then we can kind of wrap up this 1 hour already. So if you're getting antsy, by all means, feel free to leave. I don't want to hold you, but if you want to stick around for Q&A, and by all means do that as well. So what can we do with all this, and what does the road ahead look like, and how do all this techniques and all these tools kind of converge, and let us apply our expertise as engineers or physicists to and tackle interesting problems. So the problem that I'm interested in is what's called Coulomb sensing. It's a classic biophysical problem that people in the biological community have been studying for like 20 years, maybe 25 years, who knows. The first papers, the first research articles started being published in the late 90s, early 20s, mostly from Princeton, but gradually, kind of spread like fire. So the idea is, when you talk about bacteria, most bacteria are motile, meaning they can move from point A to point B using what I call flagella. And the thing about bacteria or any of these lower organisms is that when they are by themselves or when they are in small clusters, they act in ways that's very random. Or if you're more sophisticated, you'd say their behavior is stochastic. Their transport is stochastic. It does not have any order associated with it, because they don't have a central nervous system. They don't have a brain. They cannot make logical decisions the way higher organisms do, or us human beings do. They do not have individual intelligence. But once they reach a threshold number, and that number can vary based on interaction of the three F's, but whatever, once they reach a threshold number, it could be as low as 50 or as high as a few thousands, and that number is called a Coulomb. Once the Coulomb is established, once you have this minimal number, magically they start showing what's called collective intelligence, as if now this collection, this cluster of this highly non-intelligent individual units, they start showing collective intelligence. They can negotiate that microenvironment, navigate external stresses, mount elevated amounts of defense, show antibiotic resistance, be persistent to all kinds of external attacks, and create all kinds of bad infections and other pathological artifacts. That's undesirable. And they do so by kind of encapsulating themselves in this highly gooey, slimy, communal microenvironments called biofilms, which is one of the most fascinating, most mysterious advanced materials, biological materials known to humankind. We still do not understand how they form, why they form, and what goes on inside. But we do know that once biofilms form, it's exceedingly hard to get rid of them. And that's the reason we have these infections that several antibiotic attacks fail against. And people die. We have, all kinds of contamination in prosthetic devices. Catheters get clogged. All kinds of clinical settings suffer from lack of low functionality, low yield, contamination problems, infection problems because of the formation of this encapsulated structures called biofilms, which is nothing but a cluster of cells that have started secreting some kinds of complex fluids like EPS, extrapolymeric substances, which is a complicated cocktail of biological macromolecules. We do not at all understand how and when they form and what kind of communication goes inside. We do know that once you try to get rid of biofilms by attacking them with antibiotic attacks, for example, the cells kind of reorganize and self-organize in highly sophisticated patterns and subclusters and supraclusters that can negate and that can mitigate and that can cancel those external attacks. As if they talk and they really have some kind of communication between them. to reorganize in time and space as a function of external variables to kind of nullify any external threats. And it could be starvation, it could be application of thermal stresses, it could be application of extremely high pH microenvironments, but they do survive. And biofilms are some of the hardiest, most resilient microcosms known to biology. And if we understand the secrets, it helps us in two ways. We can defeat them when they are undesirable. And secondly, we can design engineered systems that show those kind of remarkable resilience in face of adversity or adverse field gradients. And that could let us design smart bio-inspired materials, which can help us from anything ranging from hazmat suits for people who work in hazardous microenvironments to highly resilient, but also highly versatile smart materials with applications in, let's say, aerospace industry, or nuclear industry, or advanced functional biomaterials. Possibilities are limitless, and I can talk more about it, but I'll say this. My recent project tries to understand how Coulomb sensing occurs in vivo inside a real microenvironment. Maybe it's kind of like doing spy work by putting your cell or putting your microscopy or imaging system inside a living, breathing, multiplying biofilm. And this is not something anybody has done before, because this is not a traditional biological question. The biological community has asked things like, signaling mechanisms, transduction, gene transcription, but they do not have the tools or the expertise or the firepower or the toolbox that somebody like me do able to ask and answer these questions. And this is where my niche, the intersection between the three F's kind of come in. So let's see what we can do about it. So we can design what's called a lab on a fiber. So take an optical fiber, and using the principles that we saw on the videos, we can actually fabricate something on the tip. So this is called a lab on a tip. So this is a highly sophisticated sensing system. And, this can generate electrical and electromagnetic signals that can talk to data acquisition systems, which can be post-processed in softwares like COMSOL Multiphysics or MATLAB to clean up the noise and draw conclusive inferences about how this thing occurs or how this phenomena happen. So what I'm trying to do here, so it's like miniaturizing all these things, the prisms, the mirrors, and the lenses, all kinds of diffraction gratings and diffraction optical elements, polarizers, and putting them on this optical tip, which is kind of the size of a human hair. And this is connected to a laser source on the other hand, and we are using an ultra-fast femtosecond laser that lasers for several billion times every second. That's incredible, but that's important for a variety of reasons. And once we send these laser signals, we can have two different community of bacteria. One sitting on the tip. We can actually have one community that we fabricate using the principles that we have talked about already. And I cannot go into a lot of details because there are a lot of details involved and we're constrained of time. But the idea is there's one community that's sitting on the tip. Let's call it community one. And there's one other community that's sitting on a optically flat surface. It could be a cover slip, like a microscope cover slip. That's sitting on an inverted microscope, kind of something like this. So this is an inverted microscope, and we have one community here, and we have an optical fiber tip, which has a community on the tip. So it's right here, if you can follow the laser pointer. And as I bring the two together and dock them, these two communities start communicating if you provide the right microenvironment. And how do they communicate? They communicate via Coulomb sensing. And now if you're smart enough, and by working with biological colleagues, I can engineer these bacteria to have a special kind of fluorophores, a special kind of genes that secrete some kind of fluorescent biomacromolecules, so-called fluorophores. And they selectively respond to certain kinds of laser wavelength, meaning they probably respond to green laser light, but not to red, while the other set responds to red laser light, but not green. And if you're smart about what kind of laser light you're shining, you can selectively see, you can selectively identify and understand the hotspots associated with the different subdomains and subregions in time and space as they are communicating. And because you have to understand this, once they communicate, or once they start communicating, the mode of transfer, they cannot send emails, they cannot shout. The mode of communication is exclusively chemical. It's by secreting and exchanging chemical molecules, which are very, very specific and highly targeted. But if they selectively respond to this laser signal that we are sending from an external high-speed, ultra-fast laser source. And they only respond selectively to some wavelengths of light. Those lights, the photons that get emitted, they can go all the way back using this duplex fiber to an optical processing system that can clean up the background noise and understand what's going on. And now we can apply this interaction of all these forces, and all these fields that we can apply on the microenvironment while maintaining these kind of flows. Sometimes these are stagnant cultures. So essentially, these are two microfluidic lab-on-a-chip bioreactors which are docking together and interacting and facilitating highly precise and highly controlled and highly targeted Coulomb sensing within highly constrained external conditions that we can precisely control and quantify and measure. This is incredible. This is incredibly powerful. And this is not something anybody has been able to do. And this is not easy. The details get really challenging and complicated, but we have made good progress. We are in the process of writing our first articles that should be published sometime later this year. But the idea is, so this was a long-winded way of saying that once you understand and appreciate the physicality issues associated with the interaction of the 3Fs at nanoscales, and once you have the tools to fabricate that in ways that's repeatable and validable and verifiable and reproducible in a way that's robust and quantifiable, you are able to integrate and interact the systems in ways that generates data sets that's impossible to generate otherwise and has been impossible to even think about in the past. And if this gets going, and if we succeed in actually quantifying this data and drawing meaningful physical conclusions from them, we can open up new gates, new vistas, new domains of how Coulomb sensing occurs at the molecular scale that empowers and instructs us and educates us about the magic and mystery associated with molecular scale chemical communication that not only helps us defeat really nasty and undesirable clinical conditions, but also lets us design highly smart, highly adaptable, and highly biomimetic engineered systems that can perform functions suited to the versatility and the multiscalarity associated with some of the new emerging fields, such as nanotechnology, bio-nanotechnology, high-performance materials, high-performance computing. The possibilities really are limitless, and you are here constrained by nothing but your imagination. This is all I got, folks. I will gratefully acknowledge the funding agencies that have been generously and graciously funded by the National Institute of Health, the National Science Foundation, the UT Southwestern Medical Center, where I was a fellow before the University of Texas System, and the Shear Labs, where I work with my mentor, Dr. Jason Shear, who's one of the pioneers in this field. So thank you all once again for attending. I really appreciate your attendance, and I would love to field any questions if you have. I hope you enjoyed this. Feel free to leave if you have to. I will stick around for as long as you guys want. So thank you once again. Pardon me, say that again, Lisa. I missed that.
We are so glad and gratified for this wonderful conclave. Indeed, it's too cool. Now we have some questions.
Absolutely. Start shooting in my way, man. I'm ready. I'm all yours.
Okay. What sparked your interest to explore this domain?
So how did I get into this? Is that what you're asking?
Yeah.
Okay, so, I didn't have any clear idea about what I wanted to do, really. So as I said, I knew right from middle school that I wanted to get a PhD and be a professor and, do science. I was never cut out for industry. So I knew I wanted to do research in some shape and form. I actually didn't want to study engineering. I hardly, so back in the days, we had this exam that no longer exists. It was called AIEEE, All India Engineering Entrance Examination. That you had to write and,
Now it is replaced by JEE Mains and JEE Advanced.
I know, it does not exist anymore. But when we attended NIT, it was via AIEEE. So I had no intentions, man, at all to, sit for it. I knew I wanted to study physics. I actually applied for the BSc Physics Honors Program at a bunch of different colleges. I got offers from the Presidency College, which is now the Presidency University, St. Xavier's College in Kolkata, and Jadavpur University and all that. And I knew that I wanted to, study physics. I was never really cut out for engineering. But, one of my uncles, he made me fill out that form. And I did not even know when the exam, the exam day was like a Sunday or somewhere. And I woke up and then my uncle was there with his bike and he was like, hey, you have this exam. And I was like, what the fudge, man? I have no exam. I need to sleep in. So I had no intentions of becoming an engineer, but I accidentally did that. So whatever, but I always wanted to do kind of physics or physics-y stuff. So I kind of fell into it. I really knew that although I'm getting an engineering degree, I'm not really cut out to work for an oil and gas industry or, for an automotive or, whatever they do these days. So after graduating college in 2010, I dabbled with a bunch of different things for the next 3 years until I started grad school. And I'm not sure how much clarity I gained about what I wanted to do then, but I really figured out what I did not want to do. So I really worked on problems that did not resonate with me, and I was like, this is not me, like this is not what I wanted to do. And typically, like some of the engineering PhDs with more engineering flavors are more about kind of optimizing. You optimize like an energy conversion system, or you make something more efficient, or you kind of have this, whatever. You have this like gas turbine, or you have this like some rotodynamic machinery, or you have this aerospace wing, and you are trying to make it more efficient, more reliable, more safe. Those kind of problems never appealed to me. I mean, it had a strong practical flavor. I was more into the fundamental side of it. So those 3 years kind of let me figure out that I wanted to do something more fundamental, and that let me decide that I wanted to go to Alberta, work at NINT, and work with Dr. Montemagno. But I still did not have much idea about what exactly I wanted to do. I'd never seen a bacteria before I started my grad school. And then, like when I started grad school, I kind of gravitated in this field. Then emerging field of biomicrofluidics. 10, 15 years ago, it was a hot new field. Now it's more established and more mature. But then I started like culturing bacteria. Oh my God, that was like super cool. This is not something I could imagine doing myself as an engineer. And that was my first exposure to the fascinating world of biophysics, or the biological engineering. And I realized that nature, as I said, Mother Nature is the ultimate, is the epitome of engineering, smarter, much more efficient, much more evolved, much more optimized than we can possibly imagine in the foreseeable future, at least. I don't know what's going to happen in several generations from now. Maybe when you guys are older, you might be able to surpass nature, but certainly not in the near horizon. And I was like, I want to study something like this. I want to study fundamental problems in nature. I want to study things with large biological flavor and interface, because I realized there's this gap in the scientific community where the traditionally trained biologists have been asking more life scientific questions like genes and signal transactions, and all these mechanisms of gene transfer, bacterial antibiotic resistance and persistence, and adaptability and evolution, and these kind of things, which is very important, don't get me wrong. But to really understand the physicality associated with biological systems, you need somebody like me. You need more people who are physically trained, people who have more quantitative skills, who can design and build devices with instrumentation capabilities, which is lacking still. Things are changing, but the landscape, I'm probably the first generation, first cohort of kind of physically trained people who are handling complicated biological questions. And I think that's cool. I think that's fascinating. The exact problem statement, you fall into it based on your lit review and funding, what kind of things you get funded for, because what I do is expensive, as you can probably guess. And at some level, it boils down to what you can manage funding for, what your funding agencies pay you for, the kind of grants you write that get funded. Sometimes there are stakeholders, or always there are, and you have to kind of like satisfy certain milestones to get the next round of funding coming. So you have to sometimes tailor it, weave the tail, and kind of spin the narrative in a way that appeals to people who are in a position of authority to give you the dollars. And you need the dollars to kind of come in at a regular basis to be able to do that. So yeah, it's a combination, primarily of this gap, this niche that I'm trying to fill in, my fascination about nature, and secondarily about more practical things like dollars and funding mandates, and what's more publishable and more maybe patentable, and what's more what are the cooler and sexier scientific topics that you can kind of like publish in good venues. And eventually I need to get a tenure track position, I need to get a job, right? So I don't want to work in something that's so pedantic, or like so airheaded it never lets you publish as well. So it's a multivariate optimization. Does that kind of answer your question?
Absolutely. Second question. Uh, Please can you elucidate some of the stories of the days you have spent in NIT Durgapur?
Oh yeah, oh absolutely. Oh my God. You don't want to open this, this is a can of monsters. I can keep going for hours. Oh my God. I had the best years of my life. So, I have since leaving NIT, I've lived in the UK, I've lived in Canada, I've lived in the United States. I've volunteered elsewhere. So I'm more traveled than the average guy in his early 30s, I guess. But I would not trade my 4 years on campus for anything else. Those were kind of like fascinating. I started, so when I was a freshman in 2006, so I was the first incoming batch for this new hall of residence for freshmen, incoming first year students. It was called Hall 9. S. Bose Hall of Residence. I don't know if it's still, a freshman.
It still exists, but now it's different. It's not in a hall for the freshmen.
Okay. Yeah, so that was exclusively like a freshman hall of residence, and it was brand new. We were the kind of the first batch of freshmen that moved in. Like it was super clean and such, which isn't the case when you go to the older halls of residences. That we realized when we were a sophomore next year. But yeah, so we kind of moved into this brand new kind of thing. Back in the days, we had hazing, so ragging was a big thing, and you could see like this big posters all over campus, stop hazing, stop ragging, all that. And if you get caught doing it as a senior or a junior, you'd be sanctioned. And if you're a freshman or a sophomore and you face it, do complain. We have zero tolerance policy. I don't know if that still exists. I know this talk is being recorded, so I don't know how honestly we can talk about it. But back in the days, it was pretty intense. And like we have these different zones as depending on where you grew up. So I'm from South Bengal. I was born and raised in the district of Purulia, in a small railway township called Adra, which is not far from the Jharkhand border. So as I'm a small city, a small town girl, a small town guy. I'm not at all. I'm not from Kolkata or I'm not from a big city. So I'm like like really one of those kind of like rural rednecks, if you will, and that was my first time living in a city, so I was like one of those like starry-eyed guys who was kind of looking at things with wonder. So like I was more gullible and more naive than the average guy from Kolkata or from bigger cities. So I was especially like targeted by my seniors when I was going through that ragging period. They called it orientation period or OP. You're not supposed to like use the word ragging because that supposedly did not exist. But oh my God, we had so many funny stories. They will call us to the senior or the junior halls of residences, like typically hall 4, hall 5, sometimes hall 3. We'll kind of go do all kinds of funny, stupid shit. Like I was terrified, but in hindsight, I mean, it was not super harmful. I mean, it can get dangerous. I certainly don't condone or endorse it in any ways, but that was a big highlight until we had our freshers welcome, and I kind of remember, people getting drunk and all kinds of things. I've never touched alcohol until that point of time. Uh, I was like super gullible. I still kind of am, I guess. And then, um, uh, yeah, that that was super cool. And then we had those like shacks, if you will, like really shitty restaurants right by the right on Mahatma Gandhi Avenue, or opposite to Samari. Uh, and they were essentially shacks that would open the doors late at night. Maybe they started serving whatever really low quality ramen noodles and like cheese omelets and such whatnot late in the nights, after midnight. They used to call them jupes. I don't know if that still exists, do they?
Yeah, it still exists.
Okay, awesome, fantastic. Oh my God. So I did not have any standards, since living abroad. I have dined in some of the best restaurants in the world. I've dined at Michelin 3-star restaurants. I've like dined at the laundry room, and Jean Georges and Donny Els and La Madeleine. But then that felt like Michelin 3-star food. It was awesome. Would hang out late at night and would pretend to study, but it was more like, gossiping, and, like they use this word PNPC, right? I cannot elaborate on that. But if there are native Bengali speakers, you can appreciate what PNPC means. We'll do that. Oh my God. I was probably more of the studious types. I was kind of nerdy and geeky. So I would study more than most of my other friends who are way smarter than I am and I was, and they will study less, but they'll still do equally well in the exams. So I probably missed out on some of the mischievous, or some of the more notorious kind of misadventures that most of the people would engage themselves in. I was probably one of the good guys, but I'll say this: I had my moments. I'll stop by saying this. By the time I had graduated, I had completely transformed from being this like gullible, like country guy, village guy, like this naive boy from someone like a confident Enitian who was able to take on the world. So that definitely wouldn't have been possible had I not hung out with the friend circle that I had for the 4 years, and the kind of crazy experiences we had while I was a student. So yeah, I don't get me started, man. I can keep talking about it for hours. But yeah, we had a lot of fun, and I would not trade that for anything else. That was awesome.
Okay. Next question. What were the challenges you faced and how did you overcome them in your
Uh,
career path?
okay. Uh, I don't know. I feel I've been really lucky and fortunate, and well, in hindsight, so I said I dabbled with a variety of research projects for 3 years right after I graduated. So most of my friends who went to grad school, they went right after immediately. They knew they wanted to get a PhD. At least a dozen of friends from my graduating class are now faculty at IITs or elsewhere. And that happened because they started grad school immediately. They went for a PhD right away. They got a PhD, they got a postdoc, they got a fellowship, they returned to India. In hindsight, it's good that I did not do that. I dabbled with a bunch of different things for 3 years. I worked at one of the IITs. I worked at IIT Kharagpur really, and I was part of this now non-existent Commonwealth Sandwich program. I was able to work with two really good mentors at the then new London Center for Nanotechnology, one at Imperial, other at UCL. And while they were really good mentors, the guy I worked with at IIT Kharagpur, and I say this off record, although I know this is being recorded, he was an absolute asshole, pardon my language. Like he was a really bad mentor, and that let me realize what I do not want to do, or what kind of people I do not want to work with, what kind of problems I wouldn't enjoy, or what would not resonate with me. While at the same time, I knew how much things are different in the West, and how much the state of the art is more advanced than what you do back at home in India. That chasm, that gap, has been shrinking over the last 15, 20 years, and while that gap was big, seemingly insurmountable, now there are several multiple research groups doing really world-class bleeding edge research in several fields right now here at India. Like there are several groups that I can actually mention who are no less than some of the top groups anywhere here in the States or in Canada or in UK or anywhere in Europe. That wasn't the case 15, 20 years ago. The federal mandates, the funding has increased a lot in the last decade or so. The public consciousness, the mindset has changed a lot. So we can certainly do world-class science in India now. That was almost an impossibility back then, and that partially motivated me to go abroad. But since I took that 3 years to kind of dabble with something, some low commitment thing, and did not commit to the rigors of a PhD program, or did not jump right into a program which I may or may not have been a good fit for, I think in hindsight that reduced or almost eliminated most of the challenges that other people face who immediately jump into that. But well, I mean, nobody cruises in grad school, so well, there are challenges meant to challenge you intellectually and, meant to challenge you professionally so that you are stretched out of your comfort zone and you kind of grow, because every growth occurs outside your comfort zone. But challenge-wise, I cannot put my fingers on something specific or something that stands out, except for there have been periods where I have been less productive or my experiments have failed and, I felt depressed, Yeah, I mean, I don't know, like I have had problems in my personal life. So when I was abroad, I lost uh, three of my grandparents. So I never had an opportunity to see my granddad from my dad's side. He had passed away even before my parents got married, so I never saw him. But my three other grandparents were alive, and I had a very deep relationship with them. While I was abroad, I lost all three within a few months, and that was kind of traumatic. I was not able to come back, attend the funerals and all that. So that was very difficult. Right after grad school in Canada, I had a girlfriend I was with for 4.5 years, and that was the woman I wanted to marry. But well, we broke up, and that was very traumatic, and those were kind of like really stressful periods for me. And I was depressed and not sure what to do. I didn't get the kind of research job that I wanted. I taught in a small liberal arts college for 1 year, McEwan University, which is very selective undergraduate. I worked as an instructor there for 1 year. But in hindsight, everything happens for good. The kind of experiences that I was able to accrue, the things I was able to add to my teaching dossier makes me a well-rounded faculty candidate, I guess. Makes me more compassionate, makes me understand things from a different lens that I otherwise not have if I did not go through the process. I actually worked in a thrift shop after my PhD. I was jobless. I eventually landed a very lucrative job with Schlumberger that I declined. But right after my PhD, for 6 months, I was working in a thrift shop, right? Minimum wage, $15 per hour. I had a PhD, but I was like showing people, they were like coming here to buy like used clothing and, like dress pants and boots and all that. And I was like showing customers around with a PhD, and I had to swallow my ego and things like that. And so that made me realize that life is not all roses, and it's not all. There would be rainy days. You have to just keep trucking and kind of like believe in something higher and trust and surrender to the universe. And if you're a good person, have good intentions and keep trying. And if you are, like not taking shortcuts, not cheating, if you're kind of committed to your cause, and if you have this higher vision, things somehow mysteriously work out in the long term. I don't understand how and why and when, and that's not really the question that you should be asking. It's not your business. Leave that to the universe or whatever higher power, consciousness, God, whatever name you like to use here. Leave it to him and kind of just be a good person, and things work out in the long term. It has over and over again when things look really dark. So yeah, I mean, I have faced lots of challenges. I wouldn't say it was like a very smooth, joyful, rosy ride. It was a very bumpy ride where my world turned topsy-turvy down several times over and over again. But I'm still alive and around and hopefully doing reasonably well and looking forward to the future. So, I mean, that's going to happen to you, every single of you, once you graduate, once you leave the sheltered intramural space of campus life, of NIT, of university, and you're on your own. You'll face challenges in some shape and form, and that makes us better human beings, or that lets us evolve and mature as people who are more compassionate and are able to contribute more faithfully and meaningfully to whatever cause that resonates with them. So be ready for challenges, but also have faith in whatever higher power you believe in, and um, surrender. Don't ask specific questions. Don't ask when and why and how. Keep doing. Be persistent. Have faith, perseverance, and persistence and patience. The three P's. Being gritty pays off. So does having faith in something that acts as an anchor and stops you from sinking. I know this firsthand because I tried. I have been in situations where I felt like I was sinking many times over and over again, but I'm still floating. So yeah, many challenges. I can keep going, but I don't know if that kind of answers your question. But I have no one single thing that stands out. It's a variety of small things that kind of get add up over time.
Indeed, too inspiring. Too inspiring. Our next question, could you Please give us some tips regarding how to proceed in research field as a guidance to those aspiring and enthusiasts to explore the research area.
Well, and once again, this is a very complicated question, and I can talk more about it, and I can talk at length about it. But well, so here's the thing. I'll tell you this. My PhD advisor, Dr. Carlo Montemagno, who is widely regarded as the founding father of bio-nanotechnology, Feynman laureate. He was the founder of what's now called the California Nanosystems Institute, one of the preeminent nanotechnology research centers in Southern California, in LA, that's now jointly managed by Caltech, UCLA, and University of Southern California. He was the founder while he was a professor at UCLA. Then he was the founder of the Nanosystems Research Group at Argonne National Lab at University of Chicago. And then he was a distinguished professor and the dean of bioengineering at Cornell before he moved to Canada to be the Canada Excellence, First Excellence Alberta Ingenuity Strategic Research Chair, where I worked with him. And guess what? If you have a PhD advisor like that, a letter from him can open doors. I mean, if Carlo writes a letter, even if it's 2 lines, and say, Sway was my grad student and he's smart, he's hardworking, I recommend him. I cannot imagine anybody not wanting to hire me. And it would have been really nice if that were happening. But guess what? Carlo is no longer with us anymore. Right after my defense, he was diagnosed with terminal stage 4 cancer, and he was gone in less than 6 months. I mean, that was traumatic. So I worked with one of the founding fathers of the field who died right after my PhD when I most needed him to write letters and provide recommendations and provide connections. He wrote a letter, an open letter on his deathbed, literally. He wrote it, had his secretary type it, and send it out to me and said, Sway, I'm leaving, but this is the letter I have written for you. It was a very generous, good letter. Share it with anybody and say, I wish you nothing but the best, but I wish I could stay here longer and, help you for more. That didn't happen. If you were here, I would probably be leading my own tenure track lab somewhere. And I don't know how much you know about this, but getting good letters is critical, is essential, is extremely crucial if you want to do good research. So my first piece of advice, which I have struggled for, since Carlo passed away in 2018, is somebody to write a freaking letter for me. I applied for a faculty position. The first thing people ask, give me a letter from your PhD advisor. And guess what? He isn't there anymore. I provide an old backdated letter, which still does the job, but it's very different from having a fresh confidential letter being sent the usual way in a sealed envelope. I'm not able to do that. People make some consideration. They kind of, they make special kind of improvisation for me. But I just wonder what would have happened if you were around and alive. So, but here's the thing, I could either cry and whine and rant about it, or I can try to make the best of whatever I can with whatever I have, which I'm trying to do. So first thing, try to build relationships with people. It could be at NIT, start with one of your favorite professors, someone who looks cool, who looks enthusiastic and dynamic, go approach him or her. I don't know if there are any ladies in mechanical engineering, and when I was a student, it was all male. As it happens, it's male dominated. Go see a professor and say, Dr. XYZ, I really like your lectures. I'm fascinated by the topics. Are you doing research or whatever? Start with that. Work on many projects here at NIT, intramural. You don't have to go to MIT to do good research. Start right at NIT. Build relationships. And understand the nuts and bolts that goes with good research. It's less about doing fancy, flashy stuff with $1 million nanofabrication equipments or transmission electron microscopy. That's all good later on, but the fundamental principles, the foundational principles behind research is all about being gritty, not giving up, having this astute eye for looking for little details, thinking out of the box and traditional, asking questions, being open to possibilities, not taking things for granted, not having these preconceived notions. These are the things that pay you well in long term. Doesn't matter where you end up, whether you are here, go to one of the IITs or like go to the States, go to Canada, go to Europe. Doesn't matter. These are basic skills. Critically thinking as a good scientist that pays you off in the long term. Try to build that even as a freshman, as a sophomore. If you're like graduating soon, try to inculcate those qualities. Talk to whoever mentors you can reach out. Reach out to people. Reach out request people to be your mentors in formal or informal capacities. And then, be strategic, be tactical, like don't chase things like brand names, or don't chase, don't chase superficial things. Find out the right fit. And I'm glad in hindsight I did that for the 3 years right after my BTech. So try to figure out something that really resonates with you. So let's say, like you have a PhD offer from MIT with a new untenured professor who's just getting started, and probably they are figuring out what they really want to do themselves, as opposed to a more established faculty member who's a fellow of the American Physical Association, or a fellow of ASME, or fellow of IEEE, in a good but non-Ivy League flagship state university like the University of Texas or University of Alberta. I would make a case that you should go to the latter and not the former, because I have seen people make these mistakes of, like chasing brand names and flashy things. But it all boils down to a good fit, whether you receive good mentorship, whether they train you and coach you in ways that lets you grow and evolve as a good scientist, because that's something that you'd be using for the rest of your career. So think long term, think from first principles, and have this intuitive conversation with you, meditate on what you really want. And by meditation, I do not mean sitting in the lotus posture, Padmasana, and doing yoga, but it means reaching this inner place of stillness where you kind of really ask and trying to figure out what you really want and what resonates with you, what engages you with the external environment, because I believe we are not humans having a spiritual experience. We are all spiritual beings having a human experience. So try to figure out that interface that really brings you alive and engages you most passionately with whatever it is that you try to pursue, or you're wanting to pursue, that craves your deepest desires. And once you know that, pursue that with full wholehearted commitment. Don't look back. Don't think about what would have happened if I had taken this job, taken that PhD offer, or gotten married, or moved to this city, or this and that. Don't ask those. And you'll receive intuitive guidance, because I believe we have this inner compass, inner GPS that lets us drops these hints and clues all the time. You just have to be receptive and tune in, and you get all this guidance all the time. You have this internal system built in. That was the most useless piece of advice probably you have heard from an alumnus, but I do not have anything more specific to say. I mean, there are other people who tell you all kinds of things, all kinds of tricks and trips about how to prepare your SOP, what kind of, how to send emails. And you'll find plenty of information about that on the internet or from other alumnus or other alumni elsewhere. My 2 cents would be just listen to yourself and reach that place of clarity, where you feel most passionately engaged. Everything else would kind of fall in place and take care of themselves. Those are little details. Once you figure out the crux and the core, the shells and the dressings and the toppings and the trimmings kind of start falling in place almost effortlessly by magic. That's the mystery, man. That's what I have realized.
Thank you so much.
Sure.
Sorry, again, I said so. Wait,
Oh,
it will be Swayamdip. Okay. Now I would ask the audience if you people had any questions. Just shoot out. Any question, guys? Please do.
Yeah, please unmute yourself and like just shoot them. I'm around. Yeah, if not, I'll stick around for maybe like 5 more minutes, and if there are no questions, then we can wrap up. But
The session had been incredible. We are indeed glad and gratified to have you, Swayamdip.
It's always an honor and pleasure to be able to speak to bright young MITians. You guys are cool. You guys are the future, and yeah, I mean, it's an honor to be able to share my 2 cents, and I hope you find something of value here. I don't know, if there was something that you felt was.
We had drawn great inspiration from you. I think that's the best thing. Drawing inspirations, gathering all these motivations. That's the thing that helps you up to build yourself as a person. So becoming a person is the most important thing, which I really feel it's absolutely needed before we can access to anything like dreams or careers. Becoming a good person, a rigid person in the sense having a rigid conscience is really
Yeah,
important.
absolutely. Yeah, have this moral compass, like you'll have these temptations to take shortcuts or, like take the easy path sometimes. Yeah, you have to like keep resisting these little temptations. And as you kind of like you keep evolving in your career and, as you kind of like reach positions of higher and higher, responsibility and authority and power, if you will, and influence, those little temptations will kind of gradually grow bigger. Don't have this moral compass and have this honesty and have this inner confidence and like have this inner sense of self-assuredness, self-security, where you are able to take the right path. And you will know, like no one has to say this. All you have to do is tune to your inner self. And as I said, because we are all human beings we are all spiritual beings having um,
Human experience.
human experience. You have access to that. You have access to that GPS navigation system. It's kind of like driving down I-35.
Exactly.
Yeah.
And at the end of the day, it will make you feel fresh. Like at the end of the day, when you have your bread at the super, you will feel super excited about life, super fresh. Uh, Even you are very tired of, messed up with all the day tasks, but being a nice person will make you fresh always.
Absolutely, yeah. If you're not feeling good, like if you know whether what you did was right or not by deciding or by kind of like noticing and gauging how you feel. If you're not feeling super good and if you're not feeling self-love, probably what you did was incorrect. So go back and fix it. Go back and kind of like, like rectify it. If you're not feeling that inner sense of pride, even if it means earning less money or being somewhere, like less flashy or, like I don't know, like passing up an opportunity which would like let you climb up the ladder faster and quicker, you still need to feel that goodness. And if you're not feeling that goodness, doesn't matter where you are, with whom you are, how much money you're making, you'd never have that satisfaction. I'm very satisfied, like I used to feel that jealousy and envy towards other people who are more successful, if you will. And once again, like success is not something you can measure, really put numbers to. But I don't anymore. I believe everybody has this trajectory, and like good things happen to you when you wait, and you're always at the right place at the right time, and whatever you need comes to you at the right time. So I am not jealous or envious anymore. Like most of my friends who never went to grad school, are making shit ton of money. Like they have bought in houses. Like they have beautiful wives. Some of them have kids. They're more secure. I'm still kind of going through this transition. Don't get me wrong, I do reasonably well for myself, but I still rent an apartment, and I still don't have a wife, or I still don't have a tenure track faculty position. So yeah, I mean, but
Since
I,
all
I
people are distinct, they will have distinct experiences. We never have the same birthdays or same time of birth. Like we are all distinct and unique by our own ways. So that's
yeah.
the thing.
Yeah, and be vulnerable, like don't try to put up this facade where, you want the world to know that you have it all figured out, or you are like bulletproof and you have everything under control. And that speaks to some level of arrogance in my opinion, because if you claim that everything is under your control, you are kind of like negating or somehow failing to acknowledge the higher supreme consciousness who knows the best and is working for your best interest. Be vulnerable and admit that, well, it's a process. You're on the trajectory. You haven't arrived, because life is not a destination really to arrive at. It's a journey. So appreciate and accept the fact, and and acknowledge that you are not perfect. You have flaws, and you have shortcomings. You have things that you're still working on, and you haven't figured it all out. And it's important to kind of like have that clarity and vulnerability, where you're not putting up a facade and being yourself. And the world sees it. The world is a game of mirrors. So if you're being vulnerable and authentic, the world responds accordingly, and good things come to you. So don't pretend to be somebody you are not, or
Right.
uh, you know don't put up faces, right? Be real, be genuine. That helps really.
And also another important thing which I deeply feel is that flaws are our distinct qualities. It's those things which make us beautiful.
Absolutely,
So
absolutely. That makes us unique as well. And you
yeah.
know And if you put up that facade of, like having it all figured out and that invincibility, you're essentially putting up the shield, this armor, like the shell that repels people. So someone who is trying to be vulnerable with you or trying to connect with you spiritually, they could be a mentor, like a next friend, next business partner, next romantic partner, whatnot. You won't let them come into your life if you're like putting up that facade. That's an armor that acts as a repellent, acts as a deterrent. Open up, vulnerable, and good things would come to you. That's my advice.
Okay, any questions from the audience side? Please, guys, don't feel afraid. Please ask some of your questions.
There are no questions. One, it was so clearly presented that you understood everything, and I doubt it because I myself do not understand the fine points. So the second conclusion is that it was so poorly presented that it went all over your head. I hope this was not the case.
No,
So
but the presentation is really incredible. Especially the videos, they're so engaging. It's obviously, the topic is something we are very much less exposed to, but still the videos were so engaging and so interesting. Like, I feel if someone has not also followed the lecture from the beginning, the videos must have left them awestruck because they were really wonderful.
Yeah, just to clarify, the videos are not mine. I provided references, so I took them from the internet because they do a really good job and way better than I could have done. So those are not my videos. Only the last section where I talked about the optical fiber off-road lab on a fiber system. That's my research. Everything else was kind of like taken from somewhere else, just to kind of give you all an idea, an overview. Because I didn't want this talk to be mathematical, I did not put a single mathematical equation. So microfluidics or nanoscale transport phenomena is extremely mathematical in nature, and there are lots of transport equations that we need to deal about to model and describe the systems. I consciously and intentionally, deliberately decided not to include any of those because I wanted to have a more general appeal and have a more kind of a bird's eye view kind of flavor for the lecture, and that's how I wanted to design it. So hopefully I did not kind of intimidate you by throwing a lot of math at you. That was intentional.
No, but we love the session entirely. It's so good. It's so nice. And especially the thing is that you are so much interactive. Like, there are many people who give us lectures or present their topics, presentations, but the most important thing is giving something like talking something about life is very important these days because these generations are moving towards a stage of frustration and depression at every moment. And I think that's very important that at the end of the day, even you feel remorse, you have someone like anyone to talk or talk about life, what life actually is, and how you really need to be. Uh, that's the most important thing, according to me.
Yeah, absolutely. Yeah, absolutely. I'm glad that you all are trying to realize that at your stage of life. I certainly did not. I was very naive and immature when I was at MIT, and like it took me a while to kind of like figure out doing adulting, for a lack of a better word. So yeah, I certainly learned it the hard way. You don't necessarily have to. I'm probably an example of, if you cannot be an example, be a warning. I don't know. But yeah, I mean, if there are no other questions, I'm not in a rush really. I probably have to head out for a brunch date with a friend in like an hour. But yeah, it's probably going to be a 15 minutes drive for me. But yeah, I can stick around for a
Actually,
few moments.
it's so we can start a little ritual from our side. I will request Minakshi to present the certificate to you. Uh, and really, I really don't have any words how to thank you because thank you. It's very little word, a two-lettered word, but I know what you have said, especially about life, experiences about life, and how to be a great person. That's really encouraging. Thank you so much, so much.
Thank you. Keep trying, being a better person, and even if that's at the cost of being a better professional engineer or a scientist or whatever. The other way around shouldn't be true. You shouldn't try to be a better scientist or a better engineer at the cost of being a better person. That's non-negotiable. The other way around is true, even if that means compromising your professional accolades and achievements a little bit. I'm not saying that you should be a slacker, but if you had to pick one, always err on the side of being a better person and being there for your friends and family, and not the other way around. I've seen way too many examples of people being burnt out, chasing false, professional dreams that never bring satisfaction. So don't be like one of those. This is very lovely. So I thank you. Really appreciate this lovely certificate. This certainly means a lot to me. And once again, it's an honor to be able to come back and speak to my brothers and sisters at NIT. So um, I'm really appreciative of this. So thank you, Meenakshi. She was, by the way, the first of contact who first.
My last question is how to engage with you through LinkedIn or any other.
Oh yeah, absolutely. So I'm on LinkedIn. I never had Facebook. As I said, like I'm kind of like I'm still a village boy in some sense, like one of those rural guys who's kind of a redneck, for lack of a better word, or like hickey, if you will. So I never had Facebook. Back in the days when I was in NIT, everybody was obsessed with the social media, the social platform called Orkut. I don't know if that still exists, but in the dorms, like in the halls of residences, everybody was on Orkut. Like creating, I don't know, communities and sending friend requests. And then it kind of started gradually transitioning to Facebook. I miss that bandwagon, man. I was never on Orkut, so I never really went on Facebook. I started straight with LinkedIn and Instagram. You are happy to connect with me on both. LinkedIn for more professional kind of thing, Instagram for more personal conversations. I am not on Facebook.
Can we have your Instagram ID? I think
Absolutely. Yeah, absolutely.
have Instagram. So
Oh, yeah, absolutely. So you can have both of that. Let me see. So I'm not super experienced with Teams. They use exclusively. It's very popular here in the States. We always use Zoom. So I haven't used Teams much. Let me see where I can access the chat. How do you access the chat?
access the chat. I think the meeting organizer can. Chandrima, can you give an access to the chat?
Maybe you have to give me some kind of access because I don't seem to have that option here. Yeah, so by all means, like I love connecting with bright young NITians like you. By all means, reach out to me on LinkedIn. If you have any questions, I will try my best to answer. So I believe Meenakshi has my official email ID. You guys can share it with anybody who wants to shoot me an email. Sometimes for more extended conversations, it's easier to have the email. I'm very happy to like chat about it. If you're in advanced stages of putting together your application, or you have multiple PhD offers that you're trying to negotiate or decide, I do not mind hopping on a Zoom call or a Teams call and kind of like sit down with you, and I can grab a glass of wine. You can like get your favorite drink, and we can like talk one-on-one at a mutually convenient time. It's 100% possible. I'd love to do that because I have received lots of guidance and mentorship from my, like people who have done this before. So I can certainly, pay that forward in my own little way. So yeah, I don't hesitate at all to reach out to me. And also I'll have my own lab sometime in fall 2023. Fingers crossed, knock on wood. I cannot give you more specific details because I'm still kind of negotiating offers and all that, so I cannot give you names. But once I do, I'll be recruiting the first set of graduate students. So if you are a senior looking to graduate and what I talked about resonates with you, and this is kind of broadly the field you want to pursue, do reach out. Obviously, I do not, I do not expect you to come join my lab. If you have better offers from more experienced PIs, by all means, go to more established groups. But if that's something if you don't mind kind of sharing the adventure of going through the process of setting up a new lab and working with a new PI who's full of ideas, by all means do reach out if that's something that resonates with you. And yeah, for any other kind of conversation, I'm very happy to I'm very happy to chat. So do reach out and don't be shy. Do connect if you like.
So can we have your Instagram ID, please? Like,
Oh,
even
sure.
we can
Unfortunately, I
connect
don't
in
don't
action.
I don't see a chat option.
So you could mail us it.
I email this to like either Lisa or Meenakshi or the email ID that the SAI Cell has, and like you
Yes,
guys
sir.
can
You
share
can you
it
can
any.
mail it to that ID.
Yeah, absolutely. Yeah, I'll certainly do that. So the LinkedIn
Thank you.
should be the primary mode for communication. Instagram is
Yeah.
more for, yeah, sure. All right. So really nice meeting you all. Good luck with whatever is ahead of you in whatever stage of career you are in, and always try to be a good person. So thank you for having me here once again. I really appreciate it. Enjoy the rest of your weekend, and have a great Saturday evening. Have fun.
Thank you,
Thank
sir.
you so much, sir.
Thank you.
Good luck to you too. Thank you. Bye bye.